So what I'm going to talk about in this Gowland Hopkins award, I will discuss in a moment or two, but I want to say a word about Gowland Hopkins. Gowland Hopkins was, of course, a scientist, a nutritional scientist of enormous eminence. He was the first professor of biochemistry at Cambridge. He was an Order of Merit. He was president of the Royal Society, and he won the Nobel Prize in 1929 in physiology or medicine for the discovery of vitamins. I never met Gowland Hopkins, of course. He died a year before I was born, but I feel that there is some sort of link in that my personal entry into nutrition, which occurred in 1975, happened because I joined the MRC Dunn Nutrition Laboratory, as Sue has already said. And the Dunn was set up very much in association with Gowland Hopkins in the 1920s. He was himself a member of the management committee of the Institute, and he gave scientific advice to the first director. So to that extent, I feel some sort of link with Gowland Hopkins. Of course, our work areas were very, very different. And again, as Sue has said in her introduction, the primary interest of my research has, in effect, been in adipose tissues, adipose tissue biology, first brown adipose tissue, and subsequently in white adipose tissue. Now, what I want to do in this presentation is to give a little bit of history, a little bit of personal chronology, and then towards the later parts of it, I want to talk about some work and some topics that have interested me, particularly in recent years. And I want to end with a proposition which you may either find provocative or obvious, and I'll leave you to choose which of those you think it actually is. Now, when I joined the Dunn in 1975, and that was the year that I joined the Nutrition Society, so I'm close to claiming to be one of the oldest and most long-standing members. And could I also say that two of the three people who nominated me, and in those days you needed 3 nominees, are currently honorary fellows of the Society, one of whom was at the lunch that we had earlier today. Now, when I joined the Dunn, it was to be a member of the energy group, which was associated with obesity that was being established by Philip James. So our concern was very much obesity. And anyone who talks about obesity has, of course, to give an example of it in one of their early slides. And this example that I've given is Sir John Falstaff, courtesy of William Shakespeare. And I put this up as my example, partly because, of course, Shakespeare was right about most things, but in this particular case, he was right in that he said, in that he has Falstaff saying, "Thou seest I have more flesh than another man, and therefore more frailty." And of course, he's absolutely right. Now, I want to show in the next couple of slides a reminder, as it were, of why obesity is regarded as a matter of some importance. And that is the simple measure of its incidence as defined by basal metabolic rate, or BMI, and this is very much the case of the UK. Now, in Shakespeare's time, obesity was present, but we assume that its incidence was really rather low. In the UK, in the early 1980s, it was also relatively low, with just 6% of adult women and 6% of adult men and 8% of adult women being clinically obese. By 2017, the most recent data that I have, we're in a situation where over a quarter of adults are now clinically obese, the figure being around 30% in the case of women. So the problem certainly has increased dramatically over a relatively short period of time. And again, as Shakespeare indicated, obesity matters because of the associated diseases, not obesity per se. And what this slide does is to summarize the major diseases that are normally associated with the obese state: type 2 diabetes, hypertension, coronary heart disease, certain cancers, etc. And it's probably type 2 diabetes that had the greatest focus. And indeed, once your BMI reaches 30, the risk of developing type 2 diabetes are increased tenfold or more, and the more obese you are, the greater that relative risk. And we're now in a situation in the UK where, according to Diabetes UK, in excess of 4.5 million people are diabetic, mostly type 2, and perhaps up to 1 million of those are undiagnosed. So it's certainly a big problem. Now, when I started in obesity research in 1975, the general view was that it was a function of gluttony and sloth. And when our group set up, we wanted to ask a very simple question: are there metabolic factors which underpin the development of obesity? And the particular role or task that I had within the energy group was to try and establish basic models using animals to investigate the issue of whether or not there were indeed metabolic differences. And the animal we used very particularly at that time was the genetically obese obob mouse. It was then the most widely used animal in obesity research, and there were two particular reasons for that. One is the scale of the obese state. After a few weeks of life, their body weight is up to threefold greater than that of lean siblings, and all that extra weight is due to fat. The second was that since this obesity is the result of a mutation in a single recessive gene, in principle, it was possible to reduce, as it were, the obese state to a simple mechanistic failure. Now, the animals that you see here, or the obese animal that you see here, is obese because, in part, it eats more. They're hyperphagic. But the question we asked at the beginning was whether or not there were indeed metabolic differences. And what we did in some of our early studies was simply to take young obese mice and remove their hyperphagia and pair feed them to the ad libitum intake of their lean siblings. And this was done by a PhD student of mine at the time, Peter Thirlby, and then after several weeks measure their energy gain. In other words, we did full energy balance studies, and what we saw was that the energy deposition in the obese animals was more than twice that of the lean, and their gross efficiency, their metabolic efficiency, as it were, was increased by a similar amount. Now, that can only occur if there's a reduction in one or more components of energy expenditure. So what were they? Now, what this slide shows is a sort of standard energy flow, energy flux diagram that we used at that time. And energy expenditure was regarded as being broken up into BMR, activity, and thermogenesis. And we were particularly attracted to the issue of thermogenesis for two particular reasons. One is that, of course, as many or some of the people here will recall, Derek Miller and Mike Stock in particular at Queen Elizabeth College in London, Queen Elizabeth as it then was, had been advocating the importance of thermogenic defects in the development of obesity. The second reason is shown in this slide here, which is a piece of data. If you take these fat obob mice and lean mice and you just put them in a cold room, a very simple, unsophisticated sort of experiment to do, what you see is that the lean mice have no problems in maintaining their body temperature. But in the case of the obese obob mouse, there is a rapid, substantial reduction in body temperature. Now, this is counterintuitive. Counterintuitive because the animals are very obese. One would think that they were extremely well insulated by the amount of body fat. So this suggests that there may indeed be an issue in terms of energy expenditure and the production of heat. And so what we then did was to measure the capacity for non-shivering thermogenesis in these animals. And what this slide shows in the grey part there is that there was no difference in energy expenditure under thermoneutral conditions, that is to say, when there is no thermoregulatory heat requirement between the lean and the obese, but when we maximally stimulated thermoregulatory non-shivering thermogenesis, as you can see here, there was a much greater response in the lean than in the obese. In other words, there's a reduced ability to generate thermoregulatory heat through non-shivering mechanisms. Now, at that time, a number of groups were reporting measurements that they'd made on lean and obese animals, metabolic measurements, enzyme levels, and so on, and finding differences. But the critical question was whether or not any differences were a consequence of being obese, or whether there was some causality associated with that defect. So we then, of course, asked that question in relation to these thermoregulatory, thermogenic abnormalities. And what we did is to take suckling of mice and lean mice, and I should emphasize that these mice become phenotypically obese at around 4 or 5 weeks of age. So we took the suckling animals at 17 days of age, and we exposed them to very mild cold, and then we put them back, having measured their rectal temperatures, marked them, and saw whether or not what happened to them once the phenotype was exhibited. And what we found here was that those that bore the lean phenotype showed a very small change in body temperature, but there was a much more marked fall in body temperature in the case of the obese. And this was true also when we did a similar experiment in 12-day-old mice. And in fact, we were able to use that as a simple means of identifying the genotype before the molecular genetics that we now have, and which, of course, we use just like that to identify matters. And what we found is that we had a very simple way of identifying the genotype before the phenotypic expression of obesity. Now, that paper was published in Nature, and I'm proud of it because it was my first nutrition-related paper. But I've also shown it up because it is quite interesting from another point of view. Those two figures and that one table are the totality of what was in that Nature paper, and the only technology we used was a fine rectal thermistor probe. Imagine sending off a paper to Nature today with just that amount of data based on temperature measurements. And I think you would count in minutes, if not in milliseconds, the speed with which the rejection email would come back. So things have changed dramatically in terms of what you need in order to get publication in the highly esteemed journals. And I'm not alone in noting that. This is a statement, and I can't remember the name of the scientist, that was published several years ago in eLife. I tell people in my lab, go and pick up a copy of Cell from the 1970s or a J-Cell biology paper. Papers that were published then would be figure 1A of a Cell paper today. So again, things have changed quite dramatically. Now, having identified differences in thermogenic ability and capacity between our lean and obese animals, the question that we then began to engage in is, what is the tissue site and the molecular mechanisms of facultative thermogenesis? And at the time that we began to engage with that question, there was a substantial growth of interest in brown adipose tissue. And what I put in this slide is a picture of the classical view of brown adipose tissue as opposed to white adipose tissue. White adipocytes are classically described as having a single large unilocular arrangement of the lipid. They have relatively sparse cytoplasm, few mitochondria, and those mitochondria are not very highly organized in terms of crystal structure. Brown adipocytes, on the other hand, have a multilocular arrangement of fat droplet, multiple fat droplets. There's an extensive cytoplasm. The mitochondria, they have large numbers of mitochondria, and those mitochondria have a very highly developed crystal structure. We, in fact, of course, now recognize that there's a third type of adipocyte, that is the bright adipocyte, which has bright or beige adipocyte, which has some of the molecular properties of brown adipocytes, but not all of them. And indeed, as it were, the field of types of adipocyte may be even more complex. Just last week, a paper in PNAS reported that there were at least 4 different subtypes of adipocyte. But I'm going to focus on the view that we had back in the late 1970s, that is adipose tissue, where adipocytes were primarily or essentially brown or white. Now, the special characteristic of brown adipose tissue is the ability to generate large amounts of heat. And this is a simple schematic of how heat is generated within the tissue. There is the release of noradrenaline from the sympathetic nerves. The tissue is densely sympathetically innervated. That noradrenaline binds to a beta-3 adrenoreceptor on the plasma membrane. There is the activation of lipolysis. Triglycerides are broken down to free fatty acids, which are oxidized in the mitochondria. But in contrast to other tissues, where the proton gradient that is normally generated across the inner mitochondrial membrane during oxidation and is linked to the synthesis of ATP, there's an alternative mechanism by which the protons move across the inner mitochondrial membrane in the case of brown adipocytes, and that is through the presence of a cell-specific, cell-unique protein, 32,000 molecular weight uncoupling protein 1. And what that essentially does is to short-circuit, in an electrical sense, the mitochondrial membrane. And it's the amount and the activity of this uncoupling protein 1 that determines the thermogenic activity and capacity of brown adipocytes. And I'm going to give one example of that in the next slide, which shows a study in which we had adapted or acclimated rats at 4 different environmental temperatures and measured by immunoassay the total amount of uncoupling protein 1 in the tissue. And I think what you can see here is that there is a stepwise increase in the amount of uncoupling protein 1, such that at 4 degrees Celsius, there is more than 100-fold increase in the total amount of that protein. So those are very dramatic changes. Now, this measurement of uncoupling protein and its activity was subsequently done in a wide variety of nutritional and physiological situations. And the main outcomes of that I've summarized in this slide. So we have a number of situations, not just cold exposure, but overfeeding. And here, the pioneering work of Nancy Rothwell and Mike Stock in the late 1970s and early 1980s should be firmly recognized. They showed that overfeeding on a cafeteria diet led to an activation of brown adipose tissue. Low-protein diets, PUFA, and so on and so forth have all been shown to be linked to an activation of brown fat. And equally, there are situations, including some of the obese mutants, and particularly the ob mouse, and that again is the pioneering work of Jean Himshagen and Michel Desautel in Ottawa in Canada. Lactation, fasting, hibernation are other examples. So by the end of the early 1980s, beginning of the 1990s, we had a clear picture of an involvement of brown adipose tissue in nutritional energetics in a wide range of situations. But the general view was that this was irrelevant to the human condition. And of course, there was a period where brown fat was being presented as the philosopher's stone in terms of the causes and as a treatment target for human obesity. Why, in retrospect, it's perhaps surprising that there was so little interest in brown fat in humans at the time, but it was essentially because people believed that humans did not contain brown adipose tissue after the first few years of adult life. And that belief was despite the work, particularly of Mike Lean at the Dunn and Daniel Riquier's group in Paris, which showed immunoactive uncoupling protein in multiple adipose tissue depots in adult humans, and also that the brown fat thermogenesis could be activated in patients with pheochromocytoma, where you get a hypersecretion of catecholamines. So by the early 1990s, my interest began to move from brown adipose tissue. It seemed, at least in human terms, to be something of a dead end. Of course, there's been a huge renaissance since 2009 with a trio of papers in the New England Journal of Medicine. Now, I changed to white adipose tissue, and I changed on December the 1st, 1994. And I changed on December 1st, 1994, when the issue of Nature that came out that day reported the identification of the OB gene, the mutant gene in OB mouse. And this gene produced a protein initially called the OB protein and subsequently called leptin. And this gene was expressed, it was thought primarily, or at the time exclusively, in white adipose tissue. And my group at the Rowett, within days, had generated probes to examine the expression of this gene. And this wasn't simply naked opportunism. 2 years earlier, a consortium of us in the UK, which included Mike Stock, had tried to get funding from several grant funding bodies in the UK to try and identify the OB gene and other mutant genes in rodents. And we were turned down on each occasion. And the reason we were given is that other groups, particularly the United States, were known to be well advanced in this, and there was no hope for us to be competitive. This was, of course, one of those very rare occasions where the rejection of one's grant application was entirely right, because there is no way that we could possibly have competed. But when we didn't get the funding, I made the strategic decision that once someone else had identified the gene, I would switch my program to chasing the physiology, as it were, of the relevant encoded protein. Well, what the discovery of leptin did was to radically change our view of the function of white adipose tissue. Of course, it had been primarily seen as a fuel storage organ. It was also believed to be associated with insulation, thermal insulation, and mechanical protection of organs, involved in glucose tolerance and inflammatory responses. But what leptin did was to identify white adipose tissue and white adipocytes as major secretory tissue and major secretory cells. And if we look at the secretory functions of white adipocytes, they're actually really very complex. Fatty acids are, of course, quantitatively the most important secretion. There are other lipid moieties that are released: cholesterol, retinol, prostaglandins, endocannabinoids, some of which are synthesized de novo within white adipocytes, others are taken up from the circulation, stored and subsequently released. Until the late 1980s, only one protein was regarded as being secreted from white adipocytes. That was lipoprotein lipase, for the uptake of fatty acids from circulating triacyl glycerols. But the discovery of leptin, as I've just said, changed it all. And not only did it change the view of adipocytes as endocrine cells, but it opened up a whole series of questions about whether or not there were other proteins that these cells might secrete. And we now have a situation where indeed there are literally believed to be several hundred proteins secreted from white adipocytes. And of course, they're given the collective name of adipokines, and just a few of them are summarized on this particular slide. They include proteins such as TNF-alpha, interleukin-1 beta, also an adipocyte hormone, adiponectin, and so on and so forth. The ones I put in red are some of the adipokines that my group at the Rowett and subsequently Liverpool identified, including zinc alpha-2 glycoprotein, nerve growth factor, and various metallothioneins. But again, we now recognize that several hundred protein moieties are actually secreted from white adipocytes. So they are really very extensive secretory cells. What white adipocytes have also become is a model system whereby questions have been subsequently asked about whether other cell types, which traditionally have never been thought of as being protein secretors, might also secrete a substantial number of proteins. And indeed, we now recognize that there are myokines from myocytes, hepatokines from liver cells, and so on and so forth. So white adipocytes have proved to be an important model for asking similar questions in other cell systems. Now, the presence of such a large, diverse number of proteins secreting from white adipocytes means that the functions and interactions of white adipose tissue with other cells and organs are really quite extensive. Clearly, through leptin, it's involved in appetite and energy balance. There are proteins associated with vascular hemostasis, lipid metabolism, blood pressure regulation, insulin sensitivity, angiogenesis, and immunity. And a substantial number are also involved in inflammation, which is why I've put that in red. And indeed, there are a whole series of inflammation-related adipokines, some of which, just a handful of which, in fact, are summarized in this slide. It includes classic cytokines and chemokines, such as TNF-alpha, interleukin-1 beta, and so on, acute phase proteins, such as PI1, serum amyloid A, and inflammation-related adipokines, such as adiponectin and matrix metalloproteinases. So we have a very extensive range of processes with which white adipocytes are involved. Now, inflammation increases in white adipose tissue as tissue mass expands in obesity, and I've shown this in this simple diagram here, which shows some of the key cytokines and chemokines whose level of expression and release increases in the obese state. And they do include TNF-alpha, interleukin-6, and so on. One particular one, adiponectin, falls, and adiponectin has insulin-sensitizing and anti-inflammatory actions. There is another component to inflammation in white adipose tissue and obesity, and that's the recruitment of macrophages. Or indeed, there are a number of different types of immune cell that are recruited into the tissue and are part of the total inflammatory state. Now, at the time of this particular report here from 2003, we began in our group in Liverpool to ask the question, well, why is it? Why should there be inflammatory response in white adipose tissue as tissue mass expands in obesity? And we came up with the following hypothesis. And that is that hypoxia occurs in the tissue as the fat mass expands, and that there are major adaptive responses to the hypoxic state involving the recruitment of the hypoxia-inducible transcription factor, HIF1, and that the recruitment of HIF1 leads to increasing expression of a series of hypoxia-responsive genes, some of which are involved in inflammation and the inflammatory response. And I should say that HIF1 is the key hypoxia-inducible transcription factor. It's made up of two subunits, HIF1 beta, which is constitutively produced, and HIF1 alpha, which is rapidly degraded in the presence of oxygen. It's stabilized as oxygen tension is low, and then you get the functional transcription factor. Now, hypoxia, of course, is traditionally associated with extreme environmental conditions such as high altitude. So why should one suggest that hypoxia might occur in white adipose tissue in the obese state? Well, the a priori reasons for so suggesting are summarized in this slide. First of all, hypoxia is known to occur in ischemic injury, during wound healing, and in solid tumors, leading to extensive metabolic changes. And indeed, certain tissues have relatively low levels of oxygen, including the retina and the brain, by way of examples. The second part, shown in red, and this is particularly from the work of Keith Frayne, last year's Blackster Award winner, that is, that blood flow to white fat is not increased in obese subjects, despite all the extra adipose tissue, and that postprandially, there is not an increase in blood flow to white adipose tissue in the obese, in contrast to the lean. And the final a priori reason was that large adipocytes are further from the vasculature than the normal diffusion distance for oxygen. A couple of years after we made that proposition, two studies came out which directly measured whether or not hypoxia does occur in white fat depots of different types of obese animal, primarily obese mouse. And some of that data is shown in this slide. On the left-hand side, there is I always struggle to say this correctly, pimonidazole staining, and Here I hope you can see there's increased staining in the case of the oddball mouse relative to the lean. If oxygen was measured directly through an oxygen electrode, then you can see on the right-hand side that there was a 2 to 3-fold difference in oxygen levels in the white adipose tissue of the obese mice. So clear, strong evidence for animal studies of hypoxia in white fat in obesity. I have to say, however, the situation is much more problematic and less clear in the case of humans. But at the time, with that data in mind, we asked ourselves the question, what are the molecular responses of adipocytes to oxygen deficiency or hypoxia? And what we did was a series of studies, which I'm going to show in the next few slides, where we took human pre-adipocytes, differentiated them into mature adipocytes in cell culture, and then we incubated them either under so-called pneumoxia, 21% oxygen, or 1% oxygen, hypoxia, for up to 24 hours, and did our measurements. And the first results of this are shown in this slide, and they indicate that there is the recruitment of HIF-1 alpha in the adipocytes under hypoxic conditions, so there is the standard response. There is also a substantial increase in the expression of the GLUT1 facilitated glucose transporter, and this is a classic marker of cellular responses to the hypoxic state. But specifically, it also suggests that there's maybe a change in glucose transport. And the next slide shows that if we measure GLUT1 protein, and the first part of this shows a standard Western blot, and its quantification below, there is in excess of a 10-fold increase in the amount of GLUT1 protein in response to the hypoxic conditions. Does this lead to changes in glucose uptake? The answer is yes. This shows the results of a 2-deoxyglucose uptake study, and here I hope you can see that in the hypoxic cells, there is a substantial increase in the amount of 2-DG uptake. And if we incubate the cells with cytokalasin B, the classic inhibitor of glucose transport, then we can see that all that hypoxia-induced increase in glucose uptake is transport mediated. And one of the consequences of this is that there is an increased lactate secretion from the cells. So we have a classic, as it were, response to oxygen deficiency with a large increase and switch on of anaerobic glycolysis. But our interest in this started in relation to adipokines. And what the next slide shows is the release or the effect of hypoxia on the release of 4 selected adipokines from the white adipocytes. And I think you can see, again, there's a large increase in leptin, VEGF, and interleukin-6 under hypoxic conditions. In contrast, there is a reduction in the amount of adiponectin. That's the protein level. If we look at gene expression, and here is a series of candidate genes, we see that essentially there are 3 types of response. Those cells which do not respond, those cells where there is an increase in gene expression, again including leptin, VEGF, and IL-6, and those like adiponectin, where there's a reduction. So there are a range of changes, but those changes at the gene level completely reflect what one would have expected on the basis of the secretion of the encoded protein. Now, this is a candidate gene approach, and what we've also done is using 43,000 probe adjuvant arrays in microarray studies, and this we did in collaboration with Dawn Mazzetti and colleagues at Unilever. And essentially what we found is that the expression of over 1,300 probes changed in 24 hours exposure to hypoxic conditions. And I should emphasize that we were quite restrictive in our criteria for a change. We said our cutoff is more than twofold change in mRNA level and p less than 0.01. You will find many a microarray study where 1.5, even 1.2-fold changes at p less than 0.05 is regarded as acceptable. So we've been quite stringent, but even with that stringency, we have over 1,300 genes whose expression has changed, roughly half of which have increased in hypoxia and half have decreased. And some of the examples which I've shown previously are also in this slide. The right-hand part of here, of course, is a standard heat map of the responses. If we look at the genes that change and do pathway analysis, then what we find at the top is that the top pathways altered by hypoxia include glucose utilization, lipolysis, lipid oxidation, cell death, and so on. Essentially what you predict from a cell where there is oxygen lack. Now, everything that I've said so far in relation to hypoxia has taken what, if you like, is the classical approach. Air, 21% oxygen versus 1%. That's what almost all hypoxia studies do. And I suppose you can say it's a proof of principle. But this is actually rather extreme, because if we look at the reality of oxygen tension, then we find that even arterial blood is below the equivalent of 15%. Tissue oxygen is roughly the equivalent of 7 to 8%. And in the case of obese mouse Watt, it's around 2%. So by taking 1 and 21, we are taking a particular extreme. And what we then did in some subsequent studies was to say, well, what happens if we use graded levels of oxygen? Do we get, as it were, a dose response or reverse dose response? Or do we find that there's a critical oxygen tension where everything is switched on by way of a hypoxic response? And I'm going to show 3 sets of data for that. We've looked at a number of adipokines. I'm going to show 2 of those. On the left-hand part of the slide is the mRNA level. On the right-hand slide is the secreted protein. And I think you can see that as we lower the oxygen tension, so there's an increase in gene expression and increased secretion of the protein, and there's no particular cutoff point. This is true not only of leptin, but here's another example. VEGF, vascular endothelial growth factor, behaves similarly too. And if we look at glucose utilization and glucose uptake, we see again a similar pattern. This is GLUT1 mRNA levels, particularly sharp change between 3 and 1% oxygen. And on the right-hand side of the slide is the change in 2-deoxyglucose uptake. And again, you can see that as we lower the oxygen tension, so we increase the response. The 2 key messages from those studies, I think. One is that there is not a critical change in oxygen tension. In effect, the cells are titrating relatively modest changes in oxygen with appropriate metabolic and molecular responses. That's the first point. The second point, and this is, I think, an issue for all of us who do and have done cell culture and tissue culture, is why do we use air? When it comes to incubation, we're meticulous in terms of pH, 7.4. We're meticulous in terms of temperature, 37 degrees, glucose concentration and other nutrients and so on, unless they happen to be the variables that we're particularly interested. But in oxygen, we seem to be perfectly comfortable to take an extreme, unphysiological situation, a situation which in effect reflects hyperoxia. Now, everything that I've said so far in relation to hypoxia relates to white adipocytes, the signature cell, as it were, of white adipose tissue. But there are, of course, several other cell types within the cell. And I just want to show the response of one other cell type, that is preadipocytes. If we look at preadipocytes, one of the early observations when leptin was discovered was that it was differentiation dependent. That is to say, that the leptin gene is not expressed when preadipocytes are taken before the induction of differentiation. It is only when they've differentiated into adipocytes that the leptin gene is expressed and leptin is released. And that RT-PCR gel shows exactly that. However, if we incubate those preadipocytes, not under normoxia, as everyone always has done, but under hypoxic conditions, what we see is that there is the induction of leptin gene expression. And if we look at this, or ask the question, is the protein secreted, then the answer is yes. There is no release of immunoreactive protein or no detectable immunoreactive leptin in the cells incubated under normoxia, but there is significant quantities of the leptin in those cells. So by incubating preadipocytes under hypoxic conditions, we can turn preadipocytes into leptin-secreting endocrine cells. There may be another message from that, of course, and that is the view that it is differentiation dependent may be wrong, because the differentiation studies are invariably done under hyperoxia, that is under 21% oxygen in air. The next slide summarizes some of the key responses to hypoxia of white adipocytes. As I have indicated, there's increased glucose uptake, increased glycolysis, anaerobic glycolysis, increased lactate release. There is a change in expression of in excess of 1,200 genes, with major changes in the release of a variety of adipokines. Other changes include a block on fatty acid oxidation, and there is also the induction of insulin resistance, which I don't have time to go into. So those are the responses to hypoxia. And what effectively we're talking about in terms of hypoxia is, in effect, deficiency of a nutrient, oxygen. In cell biology, authors will frequently refer to oxygen as a nutrient, but within nutritional science, we don't really think of oxygen as a nutrient. If you go to any textbook of nutrition, you will not find a chapter on oxygen. You will not find a section on oxygen. Indeed, any mention of oxygen will be in the context of RQ and metabolic rate. And what I suggested recently is that we should consider oxygen to be a critical nutrient and indeed a macronutrient. And this is what I say, you'll either find this as provocative or blindingly obvious. Of course, this raises a question of what is a nutrient? Well, I think we all understand what we think is a nutrient. It's something that comes in here. But if we look at the definition of it, and these are dictionary definitions, first of all, taking the Bible of the English language, the Oxford English Dictionary, a nutrient is a substance that provides nourishment essential for the maintenance of life and growth. Webster's is any substance or matter that is needed for the life and growth of living things. So on dictionary definitions, oxygen is undoubtedly a nutrient. We don't consider it a nutrient in nutritional terms, of course, for two reasons. One is the source. It's from ambient air or water in the case of aquatic species, rather than coming in through food in the diet. And secondly, of course, it enters through the lungs or gills rather than the mouth and the gastrointestinal tract. There are some other differences, of course, in that oxygen is generally abundant, although deficiency can occur, such as at high altitude. There needs to be essentially constant provision, and there's no prior processing of oxygen before it's delivered. But I would argue that we should consider oxygen as a nutrient, the forgotten nutrient. And indeed, if nutritional science wishes to be imperialistic, and I'm sure it should, then respiratory physiology should increasingly be seen as a particular subsection of nutrition. And that's my provocative bit. I want to end, of course, with some acknowledgements. And the first acknowledgements are to the students, postdocs, research fellows, colleagues and collaborators who've worked with me over the years, and they're far too many to mention. These are at a variety of institutions, and I think that Sue in her introduction indicated, and she didn't use this word, that I had been institutionally promiscuous, having worked in a number of places. And that includes the Dunn Nutrition Lab, the University of Alberta, the Rowett, in Oslo, Liverpool, and after retirement in Buckingham. And I want to particularly say how privileged I feel I am to have worked in the Dunn and the Rowett the 20th century's preeminent centres of nutritional research, not just in the UK, but internationally too. Now, those are generalized acknowledgements, and there are three particular people who I'd like to give special acknowledgements to, and they're from the early part of my career. The first on the left there is my DPhil supervisor at Oxford, Dr. Ruth van Hanegem, who gave me scientific freedom, the like of which a PhD student would not receive now, and I am eternally grateful to her for that. This particular picture is the only one I could find of her. It is celebrating her 100th birthday at St. Cross College in Oxford 2 years ago, and she's still alive. She was a founding fellow of St. Cross, and she's still with us. The second person is Philip James, who unfortunately cannot be here today. Philip took the enormous risk of recruiting me to the Dun, despite having absolutely no background in nutrition. And amongst the many qualities that he portrayed at that time was encouraging us to believe that we were doing the most important thing in the world and the most important place in the world. And I think one has never forgotten that. And the third person is David Fraser. And David Fraser nominated me for the Gowland Hopkins Award, and I'm most grateful to him for that. But David has been a colleague and a friend since I joined the Dun. He is the absolute epitome of scientific integrity and scholarship. He was a very major figure in vitamin D research, and even now, well into his 80s, he is as engaged with the details of vitamin D as he ever was, and it's a lesson for us all. There's one other person who I would like to acknowledge, and that is my wife of 50 years, and I simply want to say thank you. And thank you to the Society for the considerable honour of the Gowland Hopkins Award. Thank you.